So the next speaker will be Dr. Yves Brun from the University of Montreal. And so he is a researcher and professor in bacterial cell biology and has done a lot of things that I have difficulty understanding even. But you know, I decided to read you something from his lab page where he starts with, which I really liked. So he writes there, I have had the privilege to work with an outstanding group of people since opening of my lab in 1993. My main role is to establish the research of everyone in the lab and help them as much as I can with their subsequent careers. What I like most about my lab is the passion everyone has for understanding basic mechanisms in bacteria and the interest in helping everyone succeed. I also love cooking, hiking, and traveling, and I'm constantly in search of the perfect backpack. So that really describes, I think, an excellent supervisor. Yeah, someone you, as you are, when you're a trainee, we are looking for a place, then this is the man that you want to go to. And just one other thing, he became one of the Canada 150 research chairs. That's a program that was established in 2017 to attract great people to come to Canada to work here in Canada, and he was in Indiana at that moment. So, well.
All right, that's enough.
Yeah.
Thank you. This has been great for me because I have no background in AMR, and I also have no background in computational sciences or in artificial intelligence, and yet I'm going to talk about this stuff. One thing, since you're heading a trainee program, I've always found the word trainee kind of funny because, I'm still a trainee myself, I feel, and I learn more from the people in my lab than I'm sure they learn from me. We've heard a lot about the problem of discovering new antibiotics, and I'm glad that Dr. Lakshmi Narayan this morning ended by saying we still need new drugs. And so the problem is not that we're not getting new drugs. I mean, a few speakers have indicated that there has been an increase in the number of new drugs in the pipeline, and that's great. But the problem is that most of them are improved versions of existing drugs. And what we would like are antibiotics that that kill bacteria by mechanisms that they've never seen before. On the other hand, we have this amazing opportunity. There's this immense druggable chemical space. It's evaluated to be 10 to the 60th molecules. And so there's certain to be antibiotics in there. We just need to figure out how to find them. And so that's what I'm going to talk about. So traditionally, one of the methods that we've used to identify new antibiotics is to screen, to take a library of compounds or natural products and add them one by one to bacterial growth plates and ask who inhibits the growth of the bacteria. And that has worked well for a long time, but it has stopped producing in the last decades. And there's a few issues with this method. One is it's limited to existing libraries. Two is that existing libraries are actually in the wrong chemical space for antibiotic properties because they've been optimized for other properties that are more desirable by the pharmaceutical industry because they make money. And then it's using binary information. You have to make an arbitrary definition of what is inhibiting growth. Is it 80% inhibition? 79% good enough? And so to address the first question, a lot of groups have started using artificial intelligence to access in silico libraries. And so the way this goes is to do your screening with a library of known compounds, and then to use that information to train a machine learning model, tell the model which compounds inhibit and which compounds do not. And then the model can search a library of untested molecules and predict active molecules, and those molecules can be acquired and tested in the same way that you've generated the data. And if you get something that inhibits strongly, then you call it a hit, and you can proceed with the drug development program. And there's been some groups that have done this in recent years. It's extremely promising. We heard one version of this using peptides yesterday from Cesar de la Fuente. Here I'm talking about the small molecule space, not peptides. And so, for example, people like John Stokes at McMaster, who started recently, has published a number of interesting papers using this method. One that really floored me is a preprint that Genentech put out in September, where they screened 2 million compounds and then used that information to train a machine learning model. And they trained it with this binary information. These compounds inhibit and these do not, using a threshold of 80% inhibition. Then they used that information, that model, to screen in silico a library of 1.4 billion compounds. Enamine has about 72 billion compounds currently in their in silico library. And out of that, they got about 10,000 predicted antimicrobials. And then they selected a diversity of those, 345, and they tested them in the lab, and they got 82 compounds that had antibacterial activity, very strong antibacterial activity, and a lot of them were different than known antibiotics. So this is extremely, extremely promising. The issue is that they use this binary scoring, which is the other problem that I alluded to. And I'm gonna illustrate that problem using a simple bacterial growth curve. Right? So you start with very few bacterial cells at the beginning, and you let the cells grow either without any molecules, nutrients, but without any compounds, or with various chemical compounds, and you ask, do they inhibit? And the threshold that people have used is indicated at 16 hours. Usually people test it at 50 micromolar, and they ask for at least 80% inhibition after 16 hours. They call that a positive hit, an inhibitor, and they use that to train machine learning models. And if your results are the red and the green curve, great. That's very clear, right? But what do you do with this? Is the one that's inhibiting only at 78-ish%, is that a negative, and is the other one a positive? If you're training a machine learning model using that information and saying that the orange one is inactive, you're steering the model away from exploring that chemical space related to that molecule. What about this one? It was pretty good for a while. Then all of a sudden, it lost activity. Maybe it's unstable. And perhaps through medicinal chemistry, you can make it more stable. And so steering the model away from compounds like this is a problem. So these are just toy molecules here, but to drive this example home, if the one that loses activity after a while is the top one, and the bottom one is not in your library, you're going to get a model that will not explore that chemical space. Now, this is definitely inactive, right? I mean, the cells are growing perfectly fine with that. That's polymyxin B. This is not a high concentration, but one that is sufficient to make the cells permeable to bigger molecules. Okay, so this is with E. coli, it's gram-negative, and we just use a fluorescent dye that's too big to get through the membranes, and that stains the DNA. In the presence of polymyxin, it stains the DNA. So the point is that you need more information and not just all or nothing, a binary information. And microscopy, we think, has a really strong potential to provide rich information for training machine learning models. And I like to use a hockey analogy here. If you know a goal has been scored or not, it's binary information. It's pretty good. But if you have a picture of the goal, you have more information about it, right? You have information about the mechanism of the goal. Now, usually here I would make a joke about the Toronto Maple Leafs, but I'm going to be careful and not going to do that. And since I'm a Montreal Canadiens fan, I better not make jokes about other hockey teams. So microscopy provides you with kind of a bacterial autopsy, right? So you can get information about the effect of the molecule on the ability of bacteria to grow, and you can then train a model that can be used to screen in silico, as I've shown before. This is not a new idea. In 2013, Kit and Joe Poliano published this nice paper where they treated cells with known antibiotics, and they then stained the DNA and the membranes. And then they measured things like cell dimensions and the profiles of these fluorescent dye stainings, and they used simple principal component analysis to ask, do images of cells treated with the same compounds cluster together? And they do, as you can see here. And they also showed that if they did the same thing with a new antibacterial, for which the mechanism of action was not known, it would cluster, well, if it worked in the same way as another antibiotic, it clustered with that antibiotic, and the inference was good. You could infer the mechanism of action. So that's extremely encouraging. This approach has been used a lot recently in the pharmaceutical industry for non-infectious diseases, cancer, diabetes, you name it. And very nice work developing this cell painting approach for human cells by Ann Carpenter at the Broad and others. And some companies now make this the top of their inverted pyramid of the drug discovery process, because screening by microscopy is fast and it's not as expensive as using omics methods. And so we decided to do this for bacteria. So this summarizes about a year of work trying to figure out a good set of fluorescent dyes that will report on different things in bacteria. Eukaryotic cells have a lot of different organelles and so on, so there's rich data, but for bacteria, it wasn't clear whether we could come up with a set of fluorescent dyes that could give us rich enough information for this type of machine learning model training. And so this is the set that we've settled on for now. We stain things like DNA, the cell wall, some specific enzymatic activity, respiration, proton gradient, et cetera. And these dyes need to be spectrally compatible so that we can put them together and image them. We're currently trying to determine what is the best combination of dyes to give the best discrimination of diverse phenotypes to learn as much as we can about the effect of compounds on bacteria. And of course, if you want to do this to screen like Genentech did, 2 million compounds, you're going to need good methods for high throughput microscopy. So that's also something that we've spent some time optimizing. So our process is getting better and better. So we've settled on using 384 well imaging plates where we add the bacteria and the different compounds, and we incubate for about an hour. And that's actually good enough, as you'll see, to see differences in phenotypes, which really surprised me. Then we add the fluorescent dyes, the 5 fluorescent dyes, and then we wash and fix, and then we image. And we image using water objectives, water immersion, because it gives you a higher resolution than just air. And we think, at least that's our intuition, that higher resolution is going to be important to get better results. We can currently do about 5,000 samples a day by microscopy with our current setup. We're fully automated, robotically integrated this screening platform so that we'll have about a tenfold higher throughput, 50,000 per day without human intervention. And we're hoping that that'll be ready in about I'm hoping a year and a half, but we can already do a lot of screening with the current capability. And of course, if you get that many images, you're going to need to have good computational analysis to be able to understand how the compounds are functioning. So we've started testing this by first using a set of 4 different antibiotics that kill cells with different mechanisms of action, and DMSO, which is our negative control. And so we've just, for these results, used 2 different fluorescent stains, one for DNA and one for the cell wall, and of course we also image the bacterial cells themselves. So the first step is to identify the bacteria, right? So there's computer programs like CellPose that can identify, that can basically trace the contours of the bacteria, and then that can provide information for another program to basically measure the parameters, right? Dimensions of the cells, the fluorescent dyes, their distribution, their intensity, et cetera. And then we use simple clustering algorithms to ask which treatments look like each other, right? And this works quite well. This is clustering results. So each little dot on that plot is an individual image with a few hundred cells per image. You can see basically the colors are telling you, who's clustering there, what the model thinks is similar, and you can see that all of the images that were from treatment with A22, for example, all cluster together. Okay, so this works really well. In fact, this was done, we did this on 5 different days, and we get very good clustering when we apply a batch correction algorithm. The outliers are usually, things like this, where, dirt fell on the slide and gave a very strong fluorescent signal. So, we have very good clustering with these just 2 colors. And so next, we want to use these types of data to train a machine learning model and ask that model, can you deal with data from treatments that you haven't seen before? Okay. And so the way we do this is by first telling the model who's who. That is, these images in the training set, where we're using the same antibiotics as I mentioned before. This image was from a treatment with chloramphenicol, for example. This one was ciprofloxacin. Then that information is used to train a so-called CNN model that I'll explain in a second. And so we use most of the images for training, and then we save some for testing and validation. And then the model learns features. It learns how to interpret images of bacteria. And it extracts what it feels is important to discriminate between different treatments, and then we use clustering, as I mentioned before. So what are these CNN models? So they're convolutional neural networks. These have been designed to learn patterns from images of anything, right? You take a picture of a cat on your iPhone, it'll cluster that image with other images of cats on your iPhone, right? It's very similar conceptually, although it's not exactly the same thing. This is trained, as I said, with labeled images, so it knows, at least for the training set, what antibiotic generated this phenotype. And so it learns to recognize the important features associated with this. Then it generates a summary of the most important features for analysis. These are called embeddings. They're kind of like a book summary, basically. And then the embeddings are used for clustering, and similar embeddings are clustered together. So if I gave you a list of books like this, okay, you wouldn't have to read the full books in order to put them in categories. All you would have to read is a short summary of the books, and you could cluster them according to different types of novels. So this is basically what these models are doing. So what we did is we used this on a CNN model called ResNet 18 that's been trained on millions of images from the internet. And we trained it with this set of compounds that we've used before. And then we asked it to interpret cells treated with unseen compounds. So some that work in the same way as the ones in the training set, and some compounds that have a new mechanism of action. And we asked, is this model, has it learned well enough to be able to interpret differences between the effects of different chemicals on the bacteria? And I fully expected that we would have to optimize and fine-tune and do a lot of, as we usually do in science, before we got results that we kind of liked. But this is some of the first results that we've gotten. So this is the clustering of these 14 antibiotics from this trained model. And The boxed ones are the training set, and I'm going to point out just a few things. So A22 was part of the training set, Mycelium clusters with it. Turns out that both of these make cells a little round. Okay, so that makes sense. Then erythromycin, which the model had never seen, is clustered, is found to look similar to chloramphenicol, which they both inhibit protein synthesis. These are beta-lactams. They all cluster together because, of course, they target cell wall synthesis. These two target DNA replication. And interestingly enough, they're clustered together, but they're not clustered with ciprofloxacin, which is also an inhibitor of DNA replication. And so the model can even learn to interpret, subtleties, subtle differences between antibiotics that target similar processes but in different places. And so you can imagine if you have now a new compound in your screen and it has antibacterial activity, you ask the model to cluster it. If it works the same way as one of the things we already know, it'll cluster it with them. But for a model, for training a machine learning model to then predict compounds, what you're looking for is something that doesn't cluster with anyone. And in that case, well, how do you know how it works? What we do, I wanted to mention, I just added this, that a lot of these types of ideas can also be used for diagnostic, and the previous speaker mentioned a number of methods that seem to use microscopy as a basis for diagnostic as well. So I think a lot of this could also be applied to diagnostic. So you have a new compound that looks like it has a different effect than things you already knew. How do you know, or how can you, make your best guesses as to how it might function? So what we did is these chemical screens, but we can also do the same thing for genetic mutants. So mutants where we use CRISPR-I to basically inhibit the transcription of individual genes. And so we're doing this one by one, inhibiting the transcription of each gene in the E. coli genome, which is what we're doing currently, and we're doing the same type of microscopy to build basically a map of, how do cells behave if they don't have the activity of this protein. So if you can imagine, you have a compound now that inhibits, it's a new compound that inhibits bacterial growth, it has a new target. The phenotype might be similar to the phenotype of inhibiting the transcription of the gene. And so this is in progress. We almost have our full CRISPR-I collection spanning every gene in the E. coli genome. We're just finishing that. We've started doing phenotypic profiling of a subset of CRISPR-Is. If you went to the poster by Sahara yesterday, she has very promising results that this is actually working as we expect. And so this will really add very important information to the machine learning models to try to discover new compounds. So what we're trying to do is to build an integrated active learning-based antibiotic discovery pipeline that starts with using this microscopy, this phenomics method, in order to train a model to make predictions about potentially active compounds and then test them and make these active learning loops where the results of the experiments testing the predictions are added to the model to improve the model. The other thing that we're doing that I don't have time to describe in detail is taking a more target-specific approach. That is, we know some of the essential targets that would be good to find molecules that inhibit them, and we can use these so-called DNA-encoded libraries, which in a test tube can contain, millions of compounds, each of them with a DNA barcode, and you can add your purified protein, and you can determine just by PCR and sequencing which compounds bound to the targets. This is starting to be used a lot in industry, and we want to build these libraries in a chemical space that's more friendly to antibiotics, and we can use, again, these active learning models to determine how we're going to build these libraries. Now, we all know that the main problem is you get hits or even leads or even IND-ready compounds, and the problem is that they're toxic for humans, and that's a bad thing. So the other thing that we're doing is we want to build a toxicity model using organs on chips and metabolomics, basically treating them with different compounds and have a machine learning model learn basically what's toxic to human cells and add that information to the active learning model that will make the predictions for new compounds so that it starts biased away from toxicity, which is a good thing. So what is this magical thing, this active machine learning model that will make these predictions? It's a little different than the one that I mentioned that Genentech has just published, where those typical deep learning methods, they look for the optimal solution. And so they start going after the optimal molecules, and they forget the rest of the chemical space. The models that we're using, or that we're going to be using very soon, are called generative flow networks. Generative means that they generate new molecules. And I'll admit that with having no training in AI, I've had trouble understanding what this is about. And so I've come up with an analogy to help myself understand it. So these GFlow nets could be used to determine the best recipe for chocolate chip cookies. Okay, and this is how it would work. There's a lot of recipes out there for chocolate chip cookies, right? And so you could use what exists in the cooking literature to pre-train your model so that it can learn patterns from existing cookie recipes. And then it would generate new recipes, exploring different combinations of ingredients, different methods, etc., but not just looking for the best one right away, because there may be some that don't look quite as good, but maybe after you taste it, you get ideas about how you might modify it to get a better recipe. It samples diverse recipes, as I said, and so it doesn't just focus on the most promising one. And then it predicts recipes, we test the recipes, and we add the information to improve the model. Importantly, at all of these steps, or most of them, it uses these things called reward functions. And reward functions in this analogy is something like the predicted ease of baking, the predicted healthiness, the expected taste, the actual taste after testing. Okay. And these reward functions can be combined and weighed differently. You might want a cookie that's healthier rather than one that takes you to seventh heaven. And so you could do that with these models. So now you understand how to do this with cookies. And so just replace the word cookie with antibiotics and the words ingredients with structures or molecules, and you understand how these very complex models can help identify new antibiotics. So the next step, of course, is generate these training data. We just finished screening a 32,000 compound library that's optimized for properties that are good properties for antibiotics. And so we're going to start training models. I boxed in blue at the bottom that magic 80% inhibition that most people have used to train models. And as you can see above that, there's a lot of data points that probably point to interesting chemical space to test. The next steps are to do the microscopy screens. We're just setting up to do that. I'm hoping that this will be a Christmas present to our AI colleagues. And then we'll start training the GFlowNets models with these reward functions that are derived from both the growth and the imaging data. So we're hoping to expand the drug design into unexplored regions of this immense chemical space. And of course, this same philosophy could be applied to every step in the drug discovery pathway, right? You could have these active learning loops at every step. And this is what we would like to eventually do. This is looking far in the future, but this is where we'd like this to go. So to finish, I want to acknowledge an amazing team that we've put together for this project, both from the biology and omics side, to the AI side, to the chemistry side, And thanks for new funding by CBRIF. We're expanding this effort and we're hiring. I also want to mention people in the lab that did the work. Dave Casella is a guy who's basically led this effort. He dreamed up this whole project. It's been amazing. Zor has done the image analysis, the machine learning image analysis. Sahar was presenting a poster yesterday on the CRISPR-I. Eric has done the screening based on growth assays. James is doing the robotics optimization. And Isabelle is our strategic director. She's here. Sahar is here. And Emma is here. She had a poster also on looking at another important problem, which is dealing with the fact that gram-negative bacteria don't want to let antibiotics in because of the outer membrane. And so that's another thing that we're doing, but I don't have time to talk about it today. I'll be very happy to answer questions. Thank you very much.
Thank you, Yves. So you made us hungry with your cookie thing, so prepared us.
Yeah, that was on purpose.
So there's time for one short question and a short answer. And yeah, that's because of lunch, Yves.
Yeah. Somebody asked me if we had tested G-Flow nets with the cookie stuff, and I really want to do that. Have a nice lunch, everybody.
Yeah. Thank you.